Why don't we get started? I'm David Weinstock from the Dana Farber Cancer Institute, and welcome back to the Connect Science webinar hosted by the Dana Farber Harvard Cancer Center. It is a particular pleasure to welcome Dr. Leonard Friedman from the Frederick National Laboratory for Cancer Research. Len has, I think, more successfully than anyone I've seen, been able to straddle the academic and industry experience. He did his postdoctoral work with Keith Yamamoto, and then ran a very successful laboratory, Memorial Sloan Kettering, focused on steroid hormones, and then went off, I believe, to Merck and was at Penn and was in leadership positions at ATCC and Wyeth before landing in 2019 at Frederick National Laboratory. And I think, as Len is going to tell you, that is really a hidden gem. It shouldn't be hidden, but a remarkably robust scientific environment linked to the National Cancer Institute that provides resources for the entire cancer research community. So it's really a tremendous pleasure to hear from him. Time permitting, there will be an opportunity to ask questions. Please use the Q&A tab rather than the chat tab. And Len, again, if he has the opportunity, based on time, we'll be able to answer them during the webinar, and if not, we can connect him with the people who ask those questions, make sure they get their answers. So I'll stop there, and Len, thank you again. You're muted, sorry.
Thank you. Thank you, David. That was a very, very nice introduction, and you really did put your finger on it, and this is really the theme of my presentation today, which is that I'm going to give you, it's probably a little different than some of the talks that you've heard before in this series. I really want to present a fairly high-level overview of the depth and breadth of science that goes on at the Frederick National Lab for Cancer Research, because as David put it so accurately, it really is a hidden gem, and I try to be as objective as I can in my role as the Chief Science Officer. But to be honest with you, before I came to Frederick, I did not know there was a national lab called the Frederick National Laboratory. I knew there was the NCI site in Frederick, and I think that what I'd really like to do in the 50 or 55 minutes or so that I have today is give you a sense of the kind of work that we do here, and also with a particular emphasis on the sort of external facing initiatives that we have going that could be of use to the Harvard and Dana Farber community and beyond. And with that, I will give you a very short history. Bet you don't see a picture of Richard Nixon too often. The Frederick National Laboratory really started back in 1971 when President Nixon signed the National Cancer Act, creating the National Cancer Program and actually establishing what was originally called the Frederick Cancer Research and Development Center on 68 acres on Fort Detrick, which I'm sure you've all heard of, here in Frederick, Maryland. So an actual 68-acre sliver of land on the Army site was essentially appropriated and given to the National Cancer Institute. And in 1975, that facility was deemed a federally funded research and development center, an FFRDC. There are lots of acronyms and abbreviations in the government, and especially in the government contract world. And then in 2013, not that long ago, the Frederick NCI facility became a national lab, and the name Frederick National Laboratory for Cancer Research was given on. So we are the only federally funded research and development center, FFRDC, that's dedicated exclusively to biomedical research. You're probably more familiar with the DOE type laboratories, Livermore, Argonne, Brookhaven, et cetera. We are exclusively dedicated to biomedical research, which I'll talk to you about. I already mentioned that our major site is at Fort Detrick. Another site in Frederick is the Advanced Technology Research Facility, which is where I'm speaking to you from. You can see a picture of that below. And we have smaller sites in Bethesda on the NIH campus and Rockville. And in most of these places, and it gets a little confusing even to me, the Frederick National Laboratory employees who are contractors, we work in the public interest for Leidos Biomedical Research on behalf of the NCI. There's a co-localization of both NCI researchers and Leidos Biomedical Research employees and other contractors, just to make things even more complicated than they already are. Our mission is really to provide a unique national resource for the rapid development of new technologies. And really the key is on rapid, and I'll give you some examples of that in this talk, especially during the COVID-19 pandemic. But we're really set up as a national lab to address the most urgent and intractable problems in biomedical research and science, of course, including cancer, but also ongoing unmet challenges in HIV, and of course, threats of emerging infectious diseases. And in the last several years, those have been Ebola and COVID-19. So just to continue to give you a little bit of the flavor of the place, we have close to now 2,400 contract employees who are employed by the Frederick National Laboratory. We do provide a good deal of our work is really focused on providing operational, technical, and most especially research support to both the NCI and NIAID. We have facilities and animal maintenance at the NCI here in Frederick. We provide a lot of bioinformatics and IT support. I'll talk about the basic translational, probably not much on the clinical cancer research that we do. And as I've already mentioned, we do a lot of virus research with especially a focus on HIV, Ebola, and now SARS-CoV-2. But what I really want to talk about are the national missions, the more outward-facing initiatives that have been stood up here at the Frederick National Laboratory. And this is a partial list, believe it or not, of the national laboratory programs that have been created over the years, some several years ago, some very, very recently at FNL. I'll refer to Frederick National Lab as FNL for the remainder of this talk because it's a lot easier to say. And I'm going to try to go through each one of these with some with less detail, some with more detail. But they include nanotechnology, a very robustly resourced cryo-EM facility. I already mentioned HIV research. We have, as you probably know, the RAS initiative is based here. We have a lot of bioinformatics and data science, including a good deal of work on the Cancer Moonshot in conjunction with both the NCI and the DOE, an AI initiative I'll tell you about called ATOM, and then quite a bit of vaccine R&D, including a pilot plant that we have, again, here in Frederick, and some very important work, which I'll get into at the end of my talk, on serology, initially as it related to human papillomavirus, but now a big pivot to COVID-19. So I'm just going to start at the top and tell you a little bit about our nanotechnology characterization laboratory. That was really the first national lab initiative. It was established way back in 2004, originally as an interagency collaboration among the NCI and NIST and also the FDA. And this was really stood up as nanotechnology in basic research and certainly clinical research was really coming into its own. And a lot of people just didn't know how to use it or how to essentially take it through drug development. So really the nanotechnology characterization lab's objectives are really to provide the community expertise on characterizing nanoparticles, standardizing those methods, conducting SAR studies, facilitating regulatory reviews to those, perhaps small biotech startups or academic labs, and just generally engaging in educational knowledge and sharing efforts. So it's really a service that does the characterization for, again, academics, small biotechs, et cetera, and support to carry out clinical, regulatory, and ultimately commercialization efforts leading to hopefully approvals of therapeutics. And of course, nanoparticles are now hot again. They kind of went through a little bit of a lull, at least that was my perception. But I took this straight out of an article I read last month in Chemical and Engineering News about how, critical, of course, the mRNA vaccines from Moderna and Pfizer have been, but really without the sort of, key breakthroughs in creating the nanoparticles that the mRNA fragments were encapsulated in, we'd still be sitting and waiting for key vaccines for COVID-19. So, and that was a quote taken straight from the article. Without these lipid shells, there'd be no mRNA vaccines for COVID-19. So moving on, I did want to tell you a little bit about our cryo-EM facility. It's technically called the National Cryo-EM Microscopy Facility. It was stood up by the NCI in 2017, I believe. And of course, I don't need to tell this audience that there's been an absolute revolution in structural biology by the just incredible resolution revolution, if you will, in cryo-EM. We're now essentially at the atomic threshold in detection. The problem, of course, is that these instruments are very, very expensive. And so again, the NIH and the NCI have stood up a number of facilities, including one in this building in Frederick. And this is another slide to make the same point. New method sweeps through structural biology, but as was written last year in Science, we need a, quote, people's cryo-EM, because this is a very, very expensive tool, super powerful, but also super expensive. So the National Cryo-EM Facility's mission is to address the gap between the need for cryo-EM and the access to expensive instrumentation. As I mentioned, this facility here in Frederick opened in mid-2017, now exactly 4 years ago, with one Titan Krios microscope. A Glacios was stood up in 2018, and a second Titan Krios came online last year. And also a Japanese competitor called Joule has also installed now a fourth cryo-EM here, and it's operational here in Frederick. So we've done a little bit over 500 runs from 42 institutions across the US. And this just gives you a flavor of the institutions. But I think the important point, and back to David's introduction, is I'm not sure how many people are aware of this facility, and I'm not sure how many people that are aware of this facility are aware of the fact that it's completely free. There is no charge to anyone. You have to submit a very short description of the work. Currently, you have to prepare the grids, but we do the run and send you the data. So this just lists a number of institutions that have used the facility since it was stood up, and also towards the right here. I don't know if my cursor works here. You can see that the facility went offline for a short time when the pandemic really took off last April. And then it pivoted very quickly to COVID-related projects, which are depicted in the red. But things have gone so efficiently that we've exceeded our old goals and are now really humming along with 4 cryo-EMs here in Frederick. So I would urge everyone who doesn't have access to a facility to explore using ours. It's really as David was saying, a hidden gem here. We have a very historically, even though we're the Frederick National Lab for Cancer Research, I would say 35 to maybe now 40% of our funding comes from NIAID. And a lot of the work has historically been focused on HIV research. And this is just sort of telling you a little bit about our HIV program, which is formerly called the AIDS and Cancer Virus Program. It's led by Jeff Lifson, who's shown in the upper right here. We really provide a unique expertise and infrastructure, and that really enabled way back at the beginning of the AIDS crisis, the development of the initial HIV test kits to protect the US blood supply from spreading AIDS. I don't know how many people on this call, maybe I'm revealing my age, remember that, but that was a real challenge and a real danger at the time. The AIDS and Cancer Virus Program has also developed improved animal models, in particular to evaluate vaccines and other HIV prevention approaches. These novel animal models are being used for candidate HIV cure interventions, in particular vaccines. We've developed new tests in support of experimental treatments for infection and generated other analytical techniques and research methods. It is a true national resource. The entire HIV community really depends on the AIDS and Cancer Virus Program for all sorts of tools, reagents, and expertise. And this is just giving you one example of some of the work that has come out of the group. This is an article that was published in Nature Medicine a couple of years ago. And basically, I'm not going to go through all the data, but Jeff Lifson and his team essentially inoculated rhesus macaques with SIV, the simian immunodeficiency virus, and then initiated antiviral drug treatment at different times after inoculation. All the animals became infected. When antiviral drugs were discontinued, and this was after more than 600 days of treatment, most animals showed a rebound viremia. The virus came back and it spread systemically, which was not necessarily surprising. But what was interesting and important was that those animals that started treatment on day 5 of infection did not show this rebound viremia. So these results suggest that if virus spread can be limited during the initial virus infection, then the initial viral reservoir is presumably unstable. It can decay and potentially go to extinction. So that's a really important insight on treatment that came out of these studies. So I want to move from a little bit of the 3 initiatives that I told you and describe to you probably now the biggest National Lab initiative that has been stood up here at Frederick National Lab, and that's the RAS initiative. First of all, I'm a little reluctant to try to describe to people at the Harvard Cancer Center and Dana Farber what RAS is, but I understand that there are other people on this call, so please forgive me. I hope I'm not insulting anybody by giving a little introduction to RAS. RAS is a series of small GTPases. They're key regulators in very fundamental signal transduction pathways in both normal and cancerous cells, as this slide depicts here. As many of you know, mutated RAS is possibly the most common mutation found in human cancers. Fully 33%, a third of all human cancers have some mutated form of RAS. And until quite recently, RAS was considered undruggable. And I'll get into that in a few minutes. And that was really, I think, a challenge to many academic researchers and certainly many pharmaceutical companies. And when I was at Merck many years ago, there was a big focus on RAS, as there were at many big pharmaceutical companies, and it was summarily abandoned because RAS was so resistant to inhibitors. Oncogenic RAS, as I mentioned, is responsible for many human cancers. This digs a little bit into that in more detail. 93% of all pancreatic cancers contain a RAS mutation, 42% of all colorectal, 33% of lung cancers. It's responsible for probably more than 1 million deaths a year worldwide, and until, as I said recently, no effective inhibitors. The pathway, as I mentioned, the GTPase activity transmits a signal through RAF, MEK, ERK, and down to gene transcription. The key here is that oncogenic RAS is fully on. So this pathway, this signaling system, is never presumably off in the oncogenic state. The other, I think, reason RAS has been a challenge to study is that it is localized to the plasma membrane, and we believe a lot of its function is related to its membrane localization. And I already mentioned that RAS binds effectors, in particular RAF, to activate growth. So the RAS initiative at Frederick National Lab was started about 7 or 8 years ago, when Harold Varmus, who was at the time the director of the NCI, basically came to Frederick, saw the resources and facilities here, and said, how can the NCI apply Frederick National Lab resources to help drug the undruggable? I don't actually know if that was a direct quote, but that was the general gist of Harold's question. And if any of you know Harold Varmus, he asks these questions in a very direct way, and I can say that because he was my boss for 3 or 4 years when I was at Sloan Kettering, when he was the president of Memorial Sloan Kettering Cancer Center. And so Harold enlisted 2 key people to stand up what is now called the RAS Initiative. Frank McCormick, who is an internationally acknowledged leader in RAS biology and a professor at UCSF, and Dwight Nisley, who's been a key scientist at Frederick National Lab for a number of years. So really, the 2 of them, Frank, who before the pandemic spent a week of every month here at the Frederick National Lab, co-running the RAS initiative with Dwight Nisley. And it's been really, I think, an amazing example of what you can do with resources and a single focus. So essentially, we've stood up multiple capabilities as part of the RAS initiative. A lot of biochemistry and biophysics as it relates to KRAS membrane binding and the importance of its localization to the membrane. A lot of structural biology to reveal previously hidden pockets for inhibitor docking that until I think a few years ago really were not well known or described. Lots of assay development for KRAS effector interactions and again, compound inhibitor binding, assay development, cell-based assays, target biology, again, looking for new targets for RAS drug development, synthetic chemistry and drug discovery, computational chemistry, and the availability of RAS reagents. And again, lots of these reagents, whether they're DNA clones, proteins, cell line reagents, are supplied to external laboratories and used by the RAS initiative. So a lot of what we've set up around the RAS initiative and a few other national lab initiatives sort of emulate this hub and spoke model, where in this case the Frederick National Lab is the hub, and there are multiple spokes, extramural NCI supported laboratory collaborations, contract research with CROs, C-CRADAs with pharma and biotech, and interactions with intramural labs, particularly at the NCI. And this is another one of those, I think Frank McCormick calls it his race car slide, showing all the RAS initiative collaborations through this hub and spoke model. So, you know, there are multiple goals of the RAS initiative. I kind of view it as two sort of general goals. One is obviously to discover small molecule inhibitors that can bind to RAS and disrupt RAS effector interactions. Those would be really exciting, important, impactful drugs for cancer patients. And a number of approaches have gone into discovering inhibitors of the so-called undruggable RAS protein, which is shown on the right here. And I'll show you a couple of examples of that in a minute. And then the other is really, in some respects, more basic, but I think equally important, and that is a molecular description of RAS signaling complexes on the membrane, which is really, I think, a big challenge, and I think a lot of the tools that have been brought to bear from the RAS initiative have been very effective in, I think, unveiling some really new insights into fundamental RAS-RAF signaling, and I'll give you examples of each of these in the next few slides. No, this is the animation we all put into our slides and then forget is there. So as I said, for many years, RAS was kind of viewed as this greasy blob that had really no good docking pockets for inhibitors, and a lot of protein crystallization work has been done on RAS mutants, both here at the Frederick National Lab and elsewhere in many institutions. And what you can see here on this slide is that there are open conformations previously unknown that create reasonably large pockets that at least can be exploited for in silico screening. So on the left here, you see the pocket for the very well-studied G12C mutant. That's shown here on the left. In the center is the G13D complex with some inhibitors docked, and then finally on the right, a large pocket for Q61L is shown. So these are the kinds of structure, function, drug screening approaches that we and others have taken that I think have opened up a lot of insight. And of course, there's an Amgen compound in late clinical work. I think it's now in phase 3. So there's a lot of excitement about RAS that I don't think was generally shared, certainly a decade ago. But then there's also, as I mentioned, some really interesting fundamental biology going on in RAS-RAF signaling complexes on the membrane. And some of this really has been facilitated by molecular dynamic simulations that we've done with the the DOE laboratories as part of the Moonshot initiative. So just to sort of go back, KRAS only activates RAF kinases at the membrane. But again, the molecular mechanism of RAS activation by KRAS is essentially unknown. And so one of the questions is, could that RAS-RAF complex at the membrane be a novel therapeutic target? So there's been a lot of work done on molecular dynamic simulations that model KRAS dynamics on membrane surfaces. A lot of work done here and again in collaboration with Lawrence Livermore, Oak Ridge, Los Alamos, together with us and the NCI. And I'm just going to give you one example of that because I just think it's really cool. I'm not a particularly molecular dynamic kind of person, but you can see here that RAS is labeled with a green GFP tag, while RAF is labeled with a halo red tag. This is using total internal reflection fluorescence microscopy to enable the analysis of RAS-RAF dynamics on the inner surface of the plasma membrane. And what you can see here in this top left panel are two things. One is that RAS binds very stably. Its diffusion across the membrane is fairly slow. On the other hand, RAF is all over the place. RAF binds with a much shorter resonance time. And what it appears to be the case, if you can now look in sort of a amplification or a magnification of this panel above, is that the binding of oncogenic RAS increases the frequency of long-lived RAF molecules on the membrane. And so you can see that with that white arrow that just appeared. And when that is actually calculated, it's very, very interesting that one oncogenic RAS mutant, 4BG12D, actually engages more RAF on the membrane as observed by an increased resonance time. That's shown in red. And interestingly, and that you can compare that to wild type KRAS, which is the darker red below. And then interestingly, R89L breaks the interaction with RAF and actually doesn't even stay on the membrane. So you can see the resonance time is much shorter. So these kinds of interaction dynamics can be done with, admittedly very powerful computational tools that only a DOE national lab like Livermore or Los Alamos can provide, but it can also reveal, I think, new insights into fundamental RAS-RAF biology. So I want to move on and talk about one other computational initiative that we have going here. We have a very robust bioinformatics and data science directorate at the Frederick National Lab. A lot of that work is devoted to NCI support. But in addition to what I just showed you in the context of the RAS initiative, we're also involved in a public-private collaboration that uses AI and drug discovery called ATOM. So many of you are, or all of you are very aware, possibly with the most recent exception of our COVID-19 vaccines, that drug discovery in general is very long, very costly, and has a high failure rate. And, the question's been asked for years, and believe me, I've been in it, and I can show you some of my little scars on my body for it. We've tried all kinds of interesting approaches to shorten the timeline for R&D, whether it's preclinical to clinical. And shown in this slide is just the preclinical timeline, and could be even sort of an optimistic view of it, where lead discovery, lead optimization, and preclinical work is very linear and can take 6 years to bring something from start to finish just into the clinic. So again, there have been so many ideas and so many consulting groups that have provided the next shiny new thing to shorten this timeline that we could spend several days talking about it. But I do think that applying new concepts to computational power like AI could really lead to a significant shortening of this timeline. And so there are many, many examples of this out there. I'm acknowledging that from the get-go, but ATOM is a public-private consortium. Again, I'll describe it in a minute. It's an acronym for Accelerating Therapeutics for Opportunities in Medicine. And the idea is really to combine AI computational models, which you can see in the center here, with actual wet lab experimental work that essentially reduces that timeline that I mentioned. But there are also two other important things that I should emphasize about ATOM that may distinguish it from other initiatives. One is that the idea is that this is a public-private consortium with the goal to provide open design flow and software models that are open source design platforms made available to the research community. The original consortium are shown here. Again, I mentioned this is a public-private consortium. The founding members are UCSF, GSK, which is still part of the consortium, but not a primary member anymore. Lawrence Livermore National Laboratory really providing the high-performance computing and data science, and us, the Frederick National Lab, providing the cancer biology and data analytics. And again, as I mentioned, it's an open public-private partnership integrating data, computation, experiment, and active learning. And we want the end product is an open source framework for tools and capabilities. We, before the pandemic, actually had offices in Mission Bay, right across the street from UCSF. We have 25 FTEs engaged across the partners, the 4 partners. This was stood up in February of 2018, and we are beginning to engage new partners. So if anybody's interested on this call, please let us know. The platform is essentially described here. As I mentioned, we want to use AI-based computational models to predict and optimize molecular designs. We want to, this machine learning pipeline integrates multi-level models and system-level models, in this case, of efficacy, safety, pharmacokinetics, and drug developability to select design criteria for a new drug lead. Another interesting aspect of this is that all of these parameters, in this case, these 4 parameters, are simultaneously optimized for the generation of new molecules. And it's this multi-parameter optimization loop that really generates essentially new compounds in silico. So we create these 4 multi-level models. We run this optimization loop a number of times based on prescribed design criteria, and the output is a series of new molecules in silico. And then superimposed on that is an active learning loop that decides if and when a molecular simulation of experiment is needed to further improve or validate these models. So that goes through a molecular feature simulations, and that can also be in parallel used to set up actual experimental design, chemistry, and human relevant assay models to confirm or further inform on this discovery framework. So I'm just going to give you one example of a very early example of this multi-parameter optimization loop. And here the the machine learning pipeline prediction is really just focused on two criteria. That's the human erg that's a classic cardiovascular safety readout, and essentially aqueous solubility. And the design criteria are set at these values that are shown here, and essentially this optimization loop is run. And as you can see on the right here, active learning drives a collection of new targeted data that approaches the design criteria that were set. So with each cycle, and it goes from 0 to 30. So as we go from blue to orangish red, we are approaching that desired design criteria. Then essentially that overlaps with the green box in the upper left-hand corner. So this is an ongoing project. It's obviously been challenged like everybody else by the pandemic, but we have great partners, and this is ongoing. So the last part of the sort of guided tour of the Frederick National Lab pivots into an area that is more focused to what's going on in all of our lives today, and that is essentially vaccine R&D. And we have a very large group, which I'm not going to talk about, and I always feel very bad about this, that's devoted to clinical trials. And a lot of these clinical trials are run in very challenging locations. And in particular, our work on Ebola, which I'm not going to describe today, has been done in both Western and Central Africa under very, very, as I said, challenging conditions. But we now, thanks to the work of many collaborative groups, but including our group at the Frederick National Lab, we now have a very effective vaccine to Ebola, which has, I think, been very effective in the Central Republic of Congo, most recently. This is FNL's role in the vaccines clinical materials program, and I just bring your attention to the bottom right. We run a vaccine pilot plant that does cGMP pilot scale production in response to work that's done by the Vaccine Research Center at NIAT. And this is an example of the number of product support that has come out of our pilot plant over the last few years, from H to Z, as we say, from HIV to Zika. But what I want to talk about for the last 10 minutes is our pivot, like many other institutions, our pivot to COVID-19. And we've really pivoted in a number of ways, but generally really looking at three basic areas. And I'll give you examples of two of those in the next few minutes. One is some work by Mary Carrington, looking at the possibility of identifying genetic determinants of SARS-CoV-2 susceptibility and ultimately outcome. And the other is a very, very major effort that we have stood up here at Frederick on our expertise on serology. And I'll talk about that in a minute or two. But I'd be remiss, again, if I didn't mention a lot of work led by Beth Basler and the Clinical Monitoring Research Group on our involvement with both monoclonal antibodies and remdesivir in the treatment of COVID-19. So Mary Carrington has been at the National Lab here for a number of years, and her work has primarily been defined by incredibly important research that she published a number of years ago on the on the HLA locus. And as many of you know, the HLA locus is the most polymorphic, I think, in the genome. And in particular, her work showed that that heterozygosity of the class 1 locus associates with a slower progression of AIDS. But Mary decided when COVID-19 reared its ugly head to ask the question with multiple collaborators, does recognition of specific SARS-CoV-2 peptides by HLA result in protection? So the question is, can we identify potential HLA allotypes that associate with COVID-19 severity so that some HLA allotypes may be better able to present viral peptides, elicit a strong protection response against the virus, and therefore result in a better outcome. And Mary's been working in particular with two Boston-based institutions, Mass General and Brigham and Women's, and also UCSF and Emory. And when this slide was made, about 1,600 samples had been genotyped from patients. And of course, I don't have the results of those yet, but I wanted to mention them, as an example of how we have pivoted from, one area in this particular case, HIV, to COVID-19. So we're very excited about this work, but we'll see what the results bear out. But what I want to spend the rest of the time talking about is the serology work that we've stood up here at Frederick National Lab. And this came about in part by the CARES Act and over $300 million that the NCI got specifically to develop, validate, improve, and implement serological testing and associated technologies for COVID-19. I should mention that these were focused and distinct from the annual appropriation, so it does not take any money or resources away from cancer research. The overall program has been called the Serological Sciences Network, or SERANET, and its mission, as I said, is to increase national capacity for serological testing and advance our understanding of essentially all aspects of immune response to SARS-CoV-2 infection. So, but its features, again, I would like to believe are reflected in our ability to do this as a national laboratory. It's designed to be a very highly interactive network. It's very fast moving. There's a lot of data sharing and resources, and of course, open access publication. These are the objectives of SERANET. The biggest one, and I think thus far the most successful, was to develop and standardize novel serological assays and deploy them to the community. We've all read, very early on about antibody tests that were immediately available in the marketplace, and with no standardization, no validation, and no necessarily chance of them working in any kind of accurate way. So it was very, very important very quickly to develop and standardize novel serological tests. The other objectives are to really characterize basic biological mechanisms in response to COVID-2 and SARS-CoV-2, and to develop determined factors that modulate the immune response. So SERANET, which is really led here at Frederick by Dr. Ligia Pinto, who's the director of our Vaccine, Immunity, and Cancer program, really is, again, an example of the hub and spoke model that I described for the RAS initiative. We run the Frederick National Lab Serology Lab that Ligia is the director of, and a network coordinating center for the other external components of SERANET. And they are comprised of 13 U01 grant recipients, 8 U54 centers of excellence, and 4 capacity building centers. And again, they're designed to be very highly interactive. This is a list. I highlighted in bold the 2 Boston-based members of SERANET, Beth Israel and Harvard School of Public Health. You can see those in bold. And then there are several other UO1, U54s, and capacity building centers. The real, I think, important product, if you will, thus far that's come out of the SERANET initiative is a United States SARS-CoV-2 serology standard. And this was a joint effort involving us, the FDA, CDC, NIAID, and BARDA, and obviously SERANET academic laboratories. And so there's a large volume of pooled plasma samples were used as an assay collaborator by laboratories conducting SARS-CoV-2 testing. The main goal is obviously to harmonize those assays in measuring SARS-CoV-2 antibodies and increase comparability. That is the name of the game here, from different studies, including in response to different vaccines now. And eventually this US standard will be calibrated to the WHO international standard. It's available to SERANET academic institutions, hopefully eventually to everyone. And this is the link to download access to a small amount of this serology standard. In addition, we, again, and I should mention, let me just go back for a second, that the reason why this has been so, I think, effective and successful in terms of our ability to pivot is that prior to working on COVID-19 serology, Alicia Pinto's primary focus has been very, very important work on establishing serology standards in response to HPV vaccines, which has, just absolutely been transformational in eliminating cervical cancers. And so she was basically able to take those tools, those approaches, and very quickly pivot exactly the same kinds of approaches to SARS-CoV-2. So I say that because in addition to that pivot, our RAS initiative, which I spent quite a bit of time talking about earlier, has a very, I would say in my experience, world-class protein production laboratory, really devoted to RAS reagents. But Dom Esposito, who runs the protein production laboratory, was able to produce really beautifully purified large amounts, greater than 2 grams of COVID-19 spike RBD and nucleocapsid proteins in a very, very short amount of time, which was difficult for other laboratories to do, quite frankly, and really has been sort of a lifesaver for several initiatives, which are shown on the bottom here. I'm not going to talk about all of them, but in particular on the right panel here, his group was able to deliver COVID-19 SARS-CoV-2 encoded protein to NIAID for very critical Moderna vaccine trial serology when their supply of protein just basically dried up, as well as proteins delivered to Mali for African survey projects. So again, an example of how a national lab can pivot very, very quickly and effectively to an urgent national, or in this case, international crisis. This is my last slide. We do actively partner. We have a series of academic partnerships. They're shown here. We've had a number of appointment and exchange of scientific staff. We were doing a lot of student training like everybody prior to the pandemic. We have postdocs. We have student internships and interesting scientific projects. So with 6 minutes to spare, that is my last slide. And I really thank everybody who's on the line for their attention, and I'm happy to take questions.
Well, thank you, Len. That was really fantastic. Just a whirlwind of amazing science. There's a question. Wow, that's just kind of a larger question. Maybe I'll tailor it a little bit to it's about accessing some of these resources. So you listed a couple of websites, but I think to a certain extent, some of these things can be a little daunting. So beside maybe a form available, if I say Adam, for example, who would be the right end user, and how would that end user engage the Adam group?
Yeah, those obviously that's a great question. So there, it isn't always so simple. We have a collaborate. Probably the most effective sort of formal collaboration mechanism is through a CRADA, contractor CRADA system, and we have a partnership development office here at the Frederick National Lab. So I think the sort of first stop, depending on what the extent of the request is to go through our partnership development office. I mean, they're really, and they actually report to my office. They're really here specifically for, sort of mediating, where to go and how to go through the, whatever red tape exists. In terms of reagent exchanges, we do, transfer agreements and whatnot, but it's true. Everything is different depending on what the ask is. Adam is kind of a separate beast in itself, but it is open to collaborations, but that has to go through, Adam itself. But they have a really beautiful website, and I'd start there.
And what about for the cryo-EM?
So cryo-EM, yeah. So I would. So I wish, I should really put that link up. They have a link. They're really designed to take on quote unquote customers. So I would just, go to the National Cryo-EM Facility, NCI, Frederick, Maryland, Google it. I'm sure it'll come up and it'll be very obvious that they are a well tuned machine for taking on. I mean, that's really what they're there for.
And I'm curious, how is success defined for the RAS initiative? It's a really large and complex initiative, and you could argue that just making basic discoveries is success. But is there an essential component of translation?
That's sort of a bit of a moving target, right? I can only give you my sort of. I will only give you my view of it. I don't know. I don't know what the metric is. But I know that for a long, I'm not a RAS guy. As you mentioned, I studied nuclear hormone receptors for a long time, and they're in the nucleus. I get a little nervous when something's on the membrane. But I was certainly aware of the fact that people just left as quick. They abandoned ship as quickly as they could a number of years ago. I saw it firsthand at Merck. When it became very clear that, very expensive, very long-term, projects focused on RAS were just going nowhere. So I think that my view of success is that the fact that the field is sort of now, experienced this renaissance, and there are, dozens, if not more, scores of big pharma and biotech, focused on RAS. And there's actually there's a RAS meeting that's held that we sponsor every 2 years or every 3 years. It's coming up in the end of May, and 700-800 people will be attending that meeting, obviously virtually this year. So my sort of measure of success is that the field is back, and I think there is an Amgen compound in the clinic. There are several in the pipeline. The fact that I would like to believe that the resources of the NCI and FNL in sort of standing this whole field back up again, I think is a measure of success. And I think that our view, and again, I'm only speaking for myself, I don't want to speak for somebody like Frank McCormick, or my view is that if somebody else succeeds in actually getting a drug approved and given to patients, then we will have been successful in terms of, creating a resurgence of RAS biology.
I think that's absolutely true. And I think everyone agrees. Well, it's 1 o'clock. So I want to thank Len again for really a whirlwind lecture. Welcome everyone back next Thursday for Tom Westbrook, and then a number of fantastic lectures in the subsequent weeks. Please just check the website. And thank you again, Len.
Thank you, David. Appreciate it.